Hello, this is the first video made by the National Taiwan University about the overview of the virus. So first, let's present who are the contributors of this overview. So I'm Lucas, I come from France, I'm studying at Sciences Po Political Science and Public Administration.
Hi everyone, this is Maria Jose Carvajal from Honduras. I am an undergrad student in public health at NTU.
Hi everyone, I'm Alisa Ang from the Philippines and I'm also an undergraduate student in public health also at NTU.
Hi everyone, I'm Natasha I'm from Indonesia and I'm also an undergraduate student in public health in NTU.
So during this video, we'll present you 6 different things. First of all, the composition of the virus, then the infectious process. In the third time, we'll see the clinical picture. Then we'll see how the virus spreads. Then in a fifth part, the detection of the virus. And finally, we'll study more the case of asymptomatic patients.
So first, I would talk about the structure of the virus. So the virus is made up of 4 main proteins, namely the S protein, which is the spike glycoprotein, which has the main function of determining the direction of the virus and mediating the fusion of the virus with the host cell. The next protein is the E protein, which is also called the small envelope glycoprotein, and its function is the production and maturation of the virus. The third protein is the M protein, which is the membrane glycoprotein, and lastly we have N protein, which is the nucleocapsid protein. So the function of the nucleocapsid protein is that it's bound to the RNA. So its function is mainly for the viral genome, viral replication cycle, and viral infection. So moving on. So now moving on, we're going to talk about the infectious process. So first, the virus would enter through the ACE2 receptor of the host cell. Then the S protein would bind to the ACE2, so it would act as a receptor of the virus. Then next, the virus would be able to enter and would be able to bind through to the host cell, so it would fuse itself to the host cell. The fourth step, after they fuse, the type 2 transmembrane serine protease, or TMPRSS2, will clear all the ACE2 and activate the S protein, so it will act like it has like a spike around the infected cell, which would allow the virus to fully enter the cell. So lastly, once the virus was able to fully enter the cell, they would release the mRNA of the virus into the cytoplasm, and then it will be translated into into proteins. So it would infect the nuclei of the host cell, which would make it an infected cell.
Okay, so now we move into the clinical picture. First, we're talking about the physiological process, and we start by mentioning how the body tries to defend itself by boosting up the immune system. And this basically means that our antibodies, or in medical terms called leukocytes, move towards the lung tissues because that is the area that the virus itself targets predominantly. And then we have that on our second step, if we can call it that way, we have people having kind of an inappropriate immune response because there's a lot of inflammation going on, and even the death of epithelial cells and endothelial cells in the lung region can be observed. Then we can observe some vascular permeability being increased, and also a pulmonary edema, which is basically a medical term used for swelling. And last, in very summarized terms, we can see how it all leads to hypoxemia, which is, again, a medical term for a low level of oxygen in the blood. Elaborating further in the clinical picture, we can observe how there are systemic disorders and respiratory disorders, all of which appear and can be characterized in different time frames. We can see how the first symptoms can appear around 5 days later after the infection, And then they can last from a week to even over a month. But unfortunately, given of how new this whole experience and virus is, they are not as specific to be identified. But we can say that among symptomatic patients in the statistics that we have found, 80% present mild symptoms, while 14% present severe symptoms, and only a 6% show to appear in critical situation. And then the symptoms clearly depend by age and also on any previously existing conditions. If the patient is healthy, then it's less likely to show severity. And then we also have that there are some drugs that could be classified as non-steroidal or anti-inflammatory that could even increase the severity of the symptoms. And so, in terms of the recent findings, again, about the symptoms, everywhere in the world there have been different approaches. In Europe, we have some studies reflecting that the newest symptoms are loss of smell or even loss of taste. And then in Korea, they also support this finding. The cause of such symptoms is still uncertain, but we shouldn't take our guard down, and we should still pay attention and be alert of any new incidents or characteristics. Basically, or mainly for an earlier detection.
So after studying the part of the infectious process, it's important to raise the question of spreading, how new patients get infected. So basically the virus spreads from human to humans. There was a fear of contamination through domestic animals. However, no studies prove that those infections and contaminations from an animal to humans directly happen. So the only thing which has to be taken into account for the moment is the transmission from human to human, which happens through the droplets. So the particles coming from one human and coming into another human. Those droplets can be spread through three different means. First of all, the breath. Second of all, when someone coughs. And finally, by direct contact between two humans, or through a intermediate medium, such as paper, a phone, or any other surface. Through the droplets, mainly by breath and cough, researchers believe that the virus can spread in a distance of 1 meter 80 centimeters, which is the equivalent of 6 feet approximately. Then, concerning the transmissibility, various studies were made about that using different methods. However, there is a study which has been made recently who synthesize different studies using different methods. So this final study made by Leo is interesting because it enables us to have an overview and an average number. And according to them, the number of patients that could be infected by one infected patient is around 3.28, with a median of 2.27, which is higher than the figures given by the WHO around a month ago. So according to this study, the virus would spread more than what was expected to be few weeks ago. Then there is the question of unequal vulnerability, because not all the people are vulnerable at the same level as others. So first of all, the first category is the people who are more vulnerable than others. It's basically those who are quite old, mainly over 65 years old, and the people who had previous disease. So that can be hypertension, diabetes, cardiovascular disease, respiratory chronic disease, cancer, or being immunosuppressed. Then apparently there are some people who are less vulnerable, mainly the children and the teenagers, with the only case of a few children who happen to have the syndromes similar to the Kawasaki syndromes. However, according to the doctors, it was not the Kawasaki syndrome, it was just the same symptoms, and those few children were in very critical state. So for the moment, we don't really know why and how, but this is a point which has to be taken into account. Above all, because those cases only happen in Europe for the moment and in the US, about kids with Afro and Caribbean origins. So that has to be taken into account. Finally, there was the question of the pregnant women. Apparently, they don't tend to be more vulnerable than other people, so that's a reassuring point. Finally, in terms of vulnerability, there is the question of people having HIV. It doesn't seem that HIV itself favor the infection of COVID-19. However, because HIV comes with several comorbidities, those people could be more vulnerable. So we should give them a special care, above all concerning the access to treatment, but also concerning other issues such as mental health issues, because people with HIV tend to be more lonely than others, and the pandemic could increase these. So we should take care about this population.
Okay, so next I'm going to talk about the way of detecting the COVID-19, and the first one is the RT-PCR test. Okay, so first of all, what is it? It's a nuclear-derived method for detecting the presence of specific genetic material from any pathogen, including a virus. And how does it work? It uses a fluorescent dyes to detect targeted genetic materials, and it can be done with three types, which is nasopharyngeal swabs, throat swab, and saliva and stools. And next, I'm gonna explain about the basic steps of how RT-PCR test works. First of all, they will collect the sample from parts of the body where the virus gathers, for example, from the nose or the throat, and they will treat the sample with some chemical solutions that remove substances such as protein and fats, and extracts only the RNA present in the sample. The RNA is reverse transcribed to DNA using specific enzymes, and they will add additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA. And then the fragments attach themselves to target sections of the viral DNA if the virus is present in the sample. Next, some of the added genetic fragments are for building DNA strands during amplification, while the others are for building the DNA and adding marker labels to the strands, which are then used to detect the virus. And then the mixture is then placed in the RT-PCR machine. The machine cycles through temperatures that heat and cool the mixture to trigger specific chemical reactions that create new identical copies of the target sections of the viral DNA. The cycle will repeat over and over to continue copying the target sections of viral DNA. Each cycle doubles the previous amount. 2 copies become 4, and 4 copies become 8, and so on. As the new copies of viral DNA sections are built, the marker labels attach to the DNA strands and then release a fluorescent dye, which is measured by machine's computer and presented in real time on the screen. The computer tracks the amount of fluorescence in the sample after each cycle. When the amount goes over a certain level of fluorescence, this confirms that the virus is present. And lastly, they would also monitor how many cycles it takes to reach this level in order to estimate the severity of the infection, which is the fewer the cycles, the more severe the viral infection is. Okay, to talk about the comparison, from the three types that I mentioned before. So the first one is the nasopharyngeal swabs. As we can see, it can measure the viral RNA at the first day of the symptoms or the peak during the first week. And then, however, this swab has a limitation, which is the detection of the viral RNA does not necessarily indicate the presence of viable virus. And from here, we can see that the sensibility is between 56 to 83%, and the sensitivity is 99%. Next one is the saliva or sputum. The advantage of this one is it can be positive even when the nasopharyngeal becomes negative. And lastly is the stool. It is less probability of detection, and it appears almost at the moment of the symptoms, and also the tests stay positive on a longer period.
Next, we have the opportunity. Then, in terms of the steps, just to elaborate a little bit further on the test itself, a sample is taken, the blood is tested, and doctors particularly test for two antibodies to identify it, whether it's IgM antibodies or IgG antibodies. They have different time frames for showing up. And so the key points that we have to keep in mind are the specifics of the antibodies involved, the issue that we're dealing with, which is IgM and IgG, the timeline, or usually how long it takes for the antibody production, and then remembering that in terms of immunity, we can't know for sure if a patient is immune or not, but certainly these antibody tests can give us a good groundwork to understand what are the mechanisms found behind the blood samples that we take and whatever analysis we perform. But then the diagnostics and how you treat a patient go beyond that. Also, always keeping in mind the PCR test, which brings us to our next slide. To remember or keep in mind the difference between these two tests. For one, the coronavirus tests looks for signs of the active virus. It's simpler and faster compared to the antibody tests, but it tells you only if the virus is in your body at the moment that you're being tested. So it's like a right-away answer. Whereas the antibody tests shows you perhaps a more comprehensive understanding or overview of what you're looking at, to see whether you had the virus at some point in the past and how your body and your immune system both evolved to or responded to take care of that. The virus could be gone, or you could still be contagious, which is the case of many patients nowadays.
So this graph summarizes the different techniques that can be used to detect the virus. So we can clearly see that there are two different methods, the first one being the PCR and the second one being the antibody tests. So the PCR, as we said, tend to be relevant sooner than the antibody tests because they can be relevant even before the symptoms appear. Then we can see that several methods tend to be relevant on the longer term than others. For instance, the virus isolation from respiratory tracts tend to be relevant on the very short term. So that might not be the best method to use compared to others, such as the nasal swabs, which is the one which is the most used for the moment. And then we can clearly see that the antibody tend to appear later, and that the IgG tend to remain relevant on the longer term. So that graph really summarizes the different kind of methods and helps us to define which is the most appropriate method to use according to the test we want to make.
So now we'll be talking about asymptomatic patients. We were able to find WHO recommendations during MERS-CoV for both the people and for the health workers. So first, for the people, they recommended that if there's no other facility available to just have home isolation, but during home isolation, it's recommended that the patient who tested positive would be physically separated from anyone who's living together with them. And to, of course, follow basic hygiene rules and regular disinfection of the house. And of course, regularly check the symptoms of anyone who is in close proximity to the infected patient. And lastly, to have available information for the people just in case they have some questions or they need to consult with someone. Next would be for the health workers, of course, isolation. And then if they were tested within 24 hours and they received 2 negative tests, they should be able to go back to work. And then if the health system is in danger and needs all the health workers, they need to follow strict rules and limit the contacts to vulnerable people. With regards to the situation that was asked by Professor Wu of this asymptomatic patient who has been placed in the hospital for almost a month, we personally think that it is recommendable to make him leave the hospital. This would free up bed space and it would be less of a financial burden. However, we personally think he has to either self-isolate or preferably to go to a special facility or quarantine center, if you may call it that, for the infected people in order to monitor him or her. So next, we would just like to give suggestions for the quarantine center. For the quarantine centers, we think it would be better if you separate the quarantine centers for those who are waiting to get tested and for those who have already tested positive. So for those who have already who are waiting to be tested, these are the people who are at high risk, such as people coming from overseas or those who were in proximity to the infected patients. So these people should be separated from one another to prevent further contamination or infection. And lastly, the staff and everyone should follow strict hygiene rules to prevent spreading. This includes constant washing of hands and probably to monitor the symptoms around. So next one is for the center of the patients being tested, who has been tested positive. The reason why we think that we should separate these two is so that we could monitor those patients who have already been tested positive. So the patients that we're going to place here are those who do not require hospital care. So these are probably mostly asymptomatic patients and not really are like moderately ill or those who really don't need a constant hospital care. So first, of course, separation again of infected individuals. So this is to prevent them to be kept in enclosed spaces. So next one, the staff should not be in contact. The staff should be in contact with other centers. So we personally think that communication between quarantine centers are important. And lastly, if there's a desperate need for more healthcare workers, maybe you could ask some of the asymptomatic healthcare workers to continue working voluntarily in those centers. So this sums up our first presentation. Hopefully you were able to answer some of your questions and we're able to impart with you some useful information. If you have any questions, please don't hesitate to ask us. Thank you.
Thank
Hope to
you.
see you soon.